Good evening, everyone. As I have said you that I will be discussing the complement system or the complement cascade. So here it is. Basically, complement system is an innate immune mechanism. Like complement systems or the complements are the serum proteins, which are there to like eliminate the pathogens or the antigens and to promote inflammation. This complement system is present in all mammals, and it actually complements the action of the antibodies. So you see in the picture, like the antigen when it get entered into the blood vessels, what it does, along with the antibody, it forms the complex, which you already know that is antigen antibody complex, and this antigen antibody complex will activate the complement. So, and all under that it is written the purpose of the complement system is to remove the pathogen or the antigens. Okay. So basically in the complement system, here you will see the total 9 serum proteins, but the C1 is classified into C1Q, C1R and C1S. So total you can say 11 complement. So but actually the proteins that we are considering, the complements that we are considering is C1 to C9. So out of all the 9 complements, C3 is the one with highest concentration and C2 is the one with low concentration. Now let's move to this. These are the complements, and these are the sites where these complements are actually present, and actually it works there. It's a biosynthesis one actually. So in case of C1, it's in their intestinal epithelium, and in case of C2 and C4, which is you can say the 2 multiple. In the 2 multiple, you can see the C2 and C4, and that present in the macrophages. C3, C6, and C9, which are there in the liver, and C5, C6, C8 is in spleen, and this one C7 is not known actually. It's unknown. So you can remember this by a trick like a simple trick like the C1 will be in the intestinal one, the one that are in 2 multiple like 2 and 4, those will be in the macrophage. The one in 3 multiple, C3, C6 and C9, that will be in the liver, and the other C7 one will be unknown, and the rest which are remaining one are the C5 and C8. So these 2 complements will be there in the spleen. So in the next slide, we will be discussing about the 3 pathways of the complement system. Basically, we learned 3 pathways. So out of the 3 pathways, 2 are the most important one. That is the classical and the alternative one. So the 3 pathways are the classical activation pathway, mannose binding lectin pathway, and the third one is the alternative activation pathway. So in case of classical activation pathway, it is activated by antigen antibody immune complexes. So the trigger and the initiator is the antigen antibody immune complexes. So that will activate it. And in case of mannose binding, which is also known as mannose binding lectin, which is also known as MBL pathway, it is activated by the mannose sugar. You see in the microbes like bacteria, the cell wall of the bacteria will be having the component the mannose sugar in the cell wall, and that mannose sugar will be the activator one, the trigger one. So it will do that. And in case of alternative activation pathway, it will be activated by microbes, endotoxin, or the tumor cells. So these are the 3 pathways. We'll be discussing these 3 pathways one by one. So there are a few terminologies that we have to learn actually. 2 of them are I'm considering here because these 2 are like important in case of learning the complement pathway, and these 2 things will come up. So the first one is opsonization. So opsonization is an immune process where pathogens or dead cells are coated with proteins called opsonids, and to mark them for destruction. So in case of opsonization, what it happens, like it will coat and it will coat the pathogens, and that coating will enhance the efficiency of the phagocytosis. It will tag the antigens, and the phagocytic cells like the macrophages or the mast cells, this will come and this will identify them, and this will ingest or destroy the foreign invaders or foreign pathogens. Okay, so basically in all the three pathways, C3b, which we'll be learning in the particular individual pathways, C3b is the complement that is supposed to bind with the pathogen or the antigen that will be tagged for the phagocyte, as I have already mentioned, to destroy the pathogens. So there is a pathway you see in the factor here. In the presence of lipid carbohydrate complex, the factor C3 will get hydrolyzed to this one factor C3b, and factor C3b will combine with this factor B, and out of that factor B, with the this one convertase with the help of this factor D, the factor in case of factor B, the complement BB will be in consideration, and this C3b and BB will get combined, and the C3b and BB will be considered as the convertase one. So we can say the C3b is the complement that is supposed to bind with the pathogen. They're taking it for the phagocyte to destroy them. So you can say that opsonizer is the C3b one. It will be the one that will be doing that. So the next one is anaphylatoxin. So anaphylatoxin, here we can say the C3A, C4A and C5A. But out of these three, the C5A is the most the important one. So it will be the one which will be doing this, acting as the pro-inflammatory polypeptides, producing in the, which will be doing the activation process, activation of this complement system. So these are the things, it's written there. They trigger immune responses by binding to specific receptors. Receptors are given there, causing mast cell degranulation, increased vascular permeability, smooth muscle contraction, and chemotaxis of neutrophils. And apart from that, the anaphylatoxins, and along with that the chemotaxis also, what they do is they will promote the inflammation, and they will attract the immune cells. And as I've already told you, and it's mentioned here, the C5A is the primary activator in the all three pathways. So that's all about the two terminologies that I mentioned here. So I would like to go to now each and every pathway that I have mentioned before. All the three pathways we will be discussing in detail. So this is the first pathway that we will be discussing. This is the classical pathway. So the classical pathway, in case of the classical pathway, you see the trigger or the initiator will be the antigen and antibody complex. So antigen and antibody complex that will combine with the C1 complement. C1 will be the first complement in case of the classical pathway. And you have to also remember that antigen and antibody complex is the trigger and the initiator one. Okay, so and after that the C1 will get split. The C1 will split the C4, actually the C4 into C4A and C4B. It's already written on the right side. It will split into C4A and C4B, but apart from that the C4B will be taken into consideration. So we are taking consideration the C4B, and now C4B will cleave C2 into C2A and C2B, and C2A will remain linked to the C4B. So we are taking the 2A. We are taking the 2A complement, C2A complement, and C2B will be released into the fluid phase. So what's the new complement now? It's C4B2A, and this C4B2A is also known as CT convertase. Now you say me, why it is considered as a CT convertase? Because now this complement, the C4B2A, what it will do? It will cleave C3. The one that will cleave C3 is considered as the C3 convertase. And basically, and that's the same way the one who will be complementing the C5 will be considered as C5 convertase. Now this C4B2A, it will cleave the C3 into C3A and C3B. So this C3A is the anaphylatoxin, and C3B will be bounded with along with the C4B2A to form trimolecular complex, that is C4B2A3B, which will be considered as the C5 convertase, because in further step it will cleave C5. C5 into this is. So then it goes like C5 to C9, which is also considered as the MAC complex. MAC complex is membrane attack complex. So it will attack the membrane, and finally the cell will undergo the lysis. So this is the process here. So in case of this one also, when it will cleave the C3A, so we are considering the 3B. I forgot to mention that it will consider the C3B. So the complement that is also considered as a C5 converters will be C4B 2A 3B. Now it is considered as C5 converters because it will cleave the C5. Then this process or procedure will keep on going, but we as together we will jointly consider C5 to C9 as the MAC complex, where the membrane will get destroyed. So membrane attack complex, we consider the C5 and C9 as membrane attack complex. So this is the thing all about the classical pathway. The one thing, the few things that you have to remember is the initiator and the trigger. What is the initiator and the trigger in case of classical pathway? It's antigen and antibody complex. And what is the first complement? It's C1. What is the CT convertase here? C4B2A. And what is the C5 convertase here? C4B2A3B. So this is the thing that you have to remember. Okay. Next we'll go to the second pathway. This is mannose binding lectin or MBL pathway. So here the trigger or the initiator will be cell wall of bacteria, that is the mannose sugar. So after that what will happen? The mannose sugar will combine with the C4. Now remember here one thing, the C1, which is the first complement of this classical pathway here, so it will not be involved in case of mannose binding lectin pathway. So C1 will not be involved here, so the mannose sugar will bind with C4, which will be the first complement. So now the C4 will cleave C2 and it will convert it into C2A and C2B, but we'll consider the C2A. So the next complement will be C4B2A, so which will be C3 converters, because it will in the next step it will convert C3 into C3A C3B. So and from there we will consider the C3B. So 3B will be in the next complement will be C4B2A, and this C3B will become, okay, the 3-5 thing will come. So that will be considered as C5 converters, because in the next what you call the step, the C4B2A3B will cleave the C5 complex into C5A C5B. So but we will showing here as same like the procedure, these steps are same like the classical pathway, and it will be like C5 to C9, and that will be the MAC complex, membrane attack complex, and like that way it will, the cell will later on go and say this one lysis, or it will get destroyed. So this is the mannose binding lectin pathway. So the last one is the alternate pathway. So here it is bit different than the other two. Like here the trigger or the initiator will be the tumor cells or the endotoxins or the cancer cells, you can say. But in case of what? In case of the first classical pathway, all the complements were there from C1 to C9, everyone is involved here. In case of mannose binding lectin pathway, C1 is not involved, and in case of alternate pathway, both C1 and C2 will not be involved. So here the first complement will be C3. So the endotoxin or tumor cells will be trigger point. Now it will get this endotoxin or tumor cells will mix with this C3. It will make a complex. Then this C3 this C3 you will consider the C3B complement, and this C3B will cleave the B factor here, and from that we will take the BB, capital B small b, then the next complement will be C3B BB, which will be considered as the C3 converters, because in the further step or in the upcoming step it will cleave the C3 into C3 as C3B, and we'll consider their C3B complex or C3B1. Okay, so the C5 converters complement will be what, or the C5 converters will be what? C3B capital B small b 3B. Then the same procedure will keep on going like the previous two pathways. The C5 to this one C9, it will get this will be the membrane action. This one membrane action complex. This one will be formed, and after that membrane at the complex will form, and after that the cell lysis will happen. So these are the three pathways that we have discussed till now. So here are a few. There is a table here. So already written, the features here are given: the initiator, first complement activated, or the first complement, you can say the first complement is C3, C5 convertase, a complement level in the serum, or the immunity, in which immunity it will work on. The classical will be in the acquired, and both the lectin and the alternative one will be the innate. So till now we have studied about innate only. So in case of innate immunity, the lectin pathway and the alternative pathway will come up. So in case of initiator, in case of classical pathway, the antigen antibinding protein will come. In case of lectin pathway, the mannose binding will, mannose sugar will come. And in case of alternative pathway, the endotoxin or the cancer cell, it will come. And in case of classical pathway, the first complement will be C1. In case of lectin, it will be C4. In case of alternative, it will be C3, or you can say C3b. You see the C3 convertase here, you see the C5 convertase here. In case of this one, the complement level in serum means the all the C1 to C9, it will be in low level. Why? In case of classical pathway, because everyone is involved. So, but in case of lectin, C1 will be the normal because it is not involved, and others will be low. And in case of C1 alternative pathway, the C1, C4, and C2 will be not involved, so they will be the normal one, and others will be the low one because they are involved. So there is also a chart here in the left side top corner. So you can see the classical, what are the complements and the components that are being involved. You see the opsonization is happening, and you can see in the left side bottom corner, you can see the effects here. The opsonization, in case of opsonization, which complement will be there? The C3b. And in case of chemotoxin, it will be C5a. In case of anaphylatoxin, as I've already told you, what does anaphylatoxin do? It promotes inflammation. So the C5a will be the one which will be given the priority. The C5a will be the activator one. C5a, C3a, C4a given, but the C5a will be the activator. In case of chemotoxin and anaphylatoxin, it's the C5a only. In case of cytolysis, the C5 to C9 complements will be the one, the MAC complex, as I've told, membrane complex. And in case of enhancement of antibody production, it will be the C3b. So this is all about the complement system or complement cascade. Hope you guys have understood. So I would like to thank you all, and sorry for being late because I was due to lack of time, as I've mentioned in the previous section also. So I was not being able to make a video lecture of it, and today I have not on my face cam because of some certain reasons. And thank you all for showing the support and getting connected with me each and every day. This is the third session that we are doing about the immunology part, and I have completed the complement cascade and the complement system part. So hope you guys will like it, and hope you guys will understand fully. So I will also, as I have already linked both the PDFs and the PPTs of the previous two sections in the respective videos. So this one will be also with here. So I will attach it and I will upload it. Thank you so much.